about how useful and how what the limitations were for doing RNA-seq testing. And we felt that it was important to understand these limitations because then you can adjust for them and overcome some of the limitations. So we felt really the major question for using RNA-based technologies was that both the gene expression, which is what most people think about, but also the splicing of genes are extremely tissue dependent. So this is the most important for anyone who's interested in the brain because, as you would probably guess, it is extremely rare to get brain tissue directly to do any kind of clinical testing on. So we have postmortem tissues that you can get on a research basis, but no one is sending brain tissue for clinical testing, except in really rare exceptions. And people have published on gene expression. How do we look at what genes are expressed in the brain that are expressed in other tissues? But really, no one had done this in a comprehensive way to also look at the splicing, which is also tissue dependent. Because in practice, RNA-seq is only going to be performed on these clinical accessible tissues, which we termed cats. And when I say we termed these cats, I mean a really stellar MD PhD student in the lab, Joseph here. And this project is co-mentored by Joseph Barash, who's a computational splicing biologist who's here at Penn also. So going through, we thought, what, really we need to do a better job about not just quantifying this gene expression, but also quantifying this splicing. And so you may be thinking, oh, cats, how cute. But these are all cats from our lab. But no, these are not the cats that we're talking about. We're talking about 3 tissues that are most often used for clinical testing. So that's whole blood, lymphoblasts, which can be created from a cell line from whole blood, or occasionally fibroblasts, which can be created from a skin biopsy. So of course, whole blood is far and away the most common tissue that's used for whole blood. And so we decided to focus on these 3 tissues as the ones that are most likely to be used when someone's doing clinical testing, but really also when we're talking about the brain, these are also the most commonly used tissues for research testing as well. And so our question here was really, how can we look for the first time in a methodical way, in a quantifiable way, about how well these 3 tissues are going to do using them as a proxy, say, for the brain. So here we have an example. So if you have, this is a gene of interest that's expressed in the brain, and this particular patient is having neurologic symptoms because there's a mutation here in the gene that causes this aberrant splicing. So these are our exons, exon 1, 2, 3. These are our splice junctions when we're going from the DNA to the RNA. Here we have a mutation in the DNA that now leads to an aberrant splicing of the RNA. So this is our unknown that we are trying to find through this testing. And so what happens if we use blood to try to find this? So one of the possibilities is that there's no expression of this gene, and we are never going to find this DNA mutation if we're trying to use an RNA-based test that doesn't have expression in blood. So we remain undiagnosed. We can't find this clinical research, nothing is going to tell us about this. And that situation had been pretty well quantified. But what no one had looked at before was, what if this gene is expressed in the blood? So here we have good expression, but what happens if this splice event now, the one that's being disrupted by this mutation, isn't actually a splice event that occurs in the blood? What if this is one of the very many splice-specific isoforms that's only found in the brain? So here we're going to see great expression of this gene. However, we're not going to see any sign of this mutation. We're not going to see any sign of this aberrant RNA splicing because this exon isn't part of the isoforms that are found in the brain. So in this case, same thing, no matter how good your RNA-seq is, no matter how deeply you sequence that, you're never going to be able to find that mutation. And so here we have the only situation that we can use RNA-seq for, which is here in your blood now, you have the same splice form that you have in the brain, and you can now for the first time see the mutation using the RNA-seq. You'll be able to pick up this aberrant splicing isoform. So our question is really, we know that all three of these situations exist, but we don't know how to quantify how often each of those situations exist for any particular gene and any particular tissue. So we used a publicly available resource called GTEx, which is a tissue bank of healthy patients that right after they died, their different tissues were extracted and RNA sequencing was done on them to look at what the normal expression patterns of the RNA is in all of these tissues in normal populations. And in a very good forethought, one of the things that they also did was they also looked at whole blood, obviously, but they also made lymphoblastic cell lines from these patients, and they also made fibroblast cell lines from these patients, as if they were being taken for clinical testing or research testing. And so we took all of the data from all of these tissues. There was, it's a pretty big set of data we're working with here. It's 762 RNA-seq samples, all processed simultaneously from GTEx. That breaks down to 53 different tissues. And 3 of those are the cats, 3 of those are the clinically accessible tissues. 50 of them are everything else. It's brain, liver, stomach, everything else. And so that ends up being about 15 donors per tissue. So we get a pretty good spread of looking at different ages, different genders, different disease states and things like that. And so our criteria were we used junctions that were consistently used in the non-cats more than 50% of the time. So these are really robust splicing events. More than half of the isoforms include that splicing event. But we're not quantifiable or less than 10% of cats. So these are pretty strict criteria for saying that these are going to be important splicing junctions in the tissues that we're interested in that are really not going to be able to be found by most RNA sequencing, even pretty deep sequencing that's on a research basis. And so what did we find? So this graph, I don't expect you to be able to see the individuals. I present it more of a heat map to get a sense for the different situations that we have. And so on the y-axis, we have all the different tissues that are present in GTEx. And on the x-axis is the number of genes that fall into 3 different categories. So in the green category here, these are ones that are well represented by all 3 cats. So basically that means if something's in the green area, you could pick either whole blood, a lymphoblastic cell line, or a fibroblast cell line, and all 3 of those would pretty well recapitulate the expression and the splicing that you're seeing in the tissue of interest on the left side. If it's in the red category, that means that you have to be selective about which of the tissues that you're accessing. So you have to say the situation where you could use fibroblasts, but you can't use whole blood to recapitulate that expression and splicing. And then finally, the purple here is that it doesn't matter what you use, you're not going to find it no matter what tissue you pick. It's inadequately represented by all 3 of those clinically accessible tissues. So you can see, I'm going to get into some of the quantifications on the next slide, but you can see here that there is a little bump that we see here where we see more tissues around here that are inadequately represented by any of the accessible tissues. And that's actually because this list is alphabetical, and this bump starts with brain amygdala and ends with brain substantia nigra. And that's because this bump represents a increased number of these inadequately represented tissues that are all brain samples. So that's what this bump out of the inadequately represented tissues are. And you can see we have another bump that's down here, this line, and this, which is the most significant, where you can't like to get a peek in the splicing from any of the tissues. And this is also not surprising, this is testes. And we know that there is a ton of tissue-specific splicing that happens in the testes. So those were a nice positive control, and this ended up recapitulating what we expected based on what's known about tissue-specific splicing. So it turns out that when you quantify this, if you look across all tissues, it turns out that 40% of genes are inadequately represented by at least one cat. So if you were going to just go and pick without thinking about it, you were going to miss mutations. If you do RNA-seq and you aren't thoughtful about what tissue you miss, you're going to miss about 40% of mutations by using RNA-seq, just because, not because of any technical issues, but only because either the expression or the splicing isn't present there. And so because my students are cooler than I am and found fun memes, so this is our microarray cat detecting our transcripts on our RNA splicing here. And so so big picture, what we found, the splicing, there's about 1,300 genes for each particular non-cat tissue. So that's for the amygdala or the spleen or the testes, that's not represented by any of the cats. So none of those genes are ever going to be able to have the variants identified on RNA-seq for them. The most interesting thing that I thought from this is about 40% of those genes are not represented well, even though they're expressed. So even if you go and you look at an expression database and you say, oh, my gene is expressed in the amygdala, it should, I should find it on, it's expressed in the amygdala and it's expressed in blood, say. So if I do a blood sample, then I should still be able to see a mutation, even if I expect it to be I don't know what, an amygdala specific mutation. But even if, even if it's in the brain, say, we know that this is a gene that's important in the brain. If it's expressed in blood, even despite that, 40% of the splice variants aren't going to be there. So it's not enough to say it's just expressed there. And you might say, well, those are probably crazy genes that don't really make much difference. Maybe they're housekeeping, maybe they're only at, specific developmental time points, so that it doesn't really make a difference. So because we're interested in this and helping clinical and translational work, we went back and said, well, those specific genes, how many of them are actually important for human disease? And it turns out that just over 30% of those genes have already been annotated as disease-causing genes. So it seems that these are at least represented, if not overrepresented, by genes that have clinical importance. Actually, I should update that because the paper is now available. So this, if you want to learn more about this, it's in Genetics and Medicine, just came out recently. And so some people, say, well, I have all this RNA-seq data, I'm not really sure about it. If you do want to collaborate with anything that has to do with RNA-seq data, especially, splicing, what might be missing, different, we'd be happy to help plan RNA-seq experiments based on this data. Just reach out to us. Most importantly, so everyone, at this point in the talk, everyone says, well, what was the best tissue? Oh, are there, sorry, checking chats. Yes, oh, yeah, sorry. So everyone says, well, what was the best tissue? What should we use? And it's a little bit difficult to say that as a blanket statement. So it turns out that generally, if you have no idea, you should do fibroblasts. But fibroblasts are, of course, much harder to obtain than blood. And then the other thing that makes it a little funny to make generalizations about is the blood that was used in GTEx wasn't hemoglobin depleted. So a lot of the transcripts that you get from whole blood are just boring hemoglobin, and we don't care about that. And so that throws off the results a little bit. But we had enough people asking these questions and emailing us that we used the help of Paul Jewell, who's a programmer at Penn, who turned all of the resources that Joseph had put together and actually made it a publicly available interactive online resource. And so you too could go to this website, tools.biocyphers.org, MagicCat. Magic is the name of the splicing tool that we use for splicing prediction. And you can type in, you can see, I pulled up here for you guys a screenshot of the brain. You can pull up all brain, specific brain subsections of the brain. You can pull in, I want only ClinGen annotated disease-causing genes. I want, what's some of the deciphering developmental disabilities disorder, brain important genes. You can pull in custom lists. You can say, I want all epilepsy genes. Really, you know, this is, it's, I am not a computer person, and it's very user-friendly. So, so if you, you know, please, if you have any suggestions for using that, or we'd love to hear them back. But it's great for writing grants if you're doing anything that has to do with RNA, because either you can prove that you can use blood or fibroblasts to it, or you can justify getting extra tissues to an IRB or someone that might give you difficulty for getting non-cat tissues on a research basis. And so just to give you an example, this is the type of, this is the output that we get. So it'll tell you both the tissue expression for the particular genes. I pulled up one of my favorite brain genes, MEF2C. And so it'll give you a violin plot for the expression, and also the splicing, how well that's represented in your cat. So you can see for this one, you are not gonna, this splice event doesn't occur in EBV transformed lymphoblastic cell lines, but we do see the splicing event. So this is a splicing event that's important in the cortex of the brain. It's important in the fetal cortex of the brain, but it's not present in the whole blood or the EBV transformed cells. And so you would completely miss any mutations in this, even though it's clearly important in brain development. But even though this gene is expressed, this particular splice variant wouldn't come up. So let me pause there, because that's kind of one particular story chunk. Does anyone have any questions about MagicCat or RNA-seq splicing? Anything that would be interesting to talk about before we move on to the next session? And I'm gonna.
I had one quick question, if that's all right.
Yes, of course, definitely.
Just in regards to the tissue-specific expression in MagicCat, I saw a lot of brain-specific options. Does it cover extracellular RNA from CSF? So, for example, lumbar puncture. Could you apply that to that, or is that not available from GTEx? I'm not familiar.
Oh, that's a great question. So I am about 95% sure that that is not a GTEx tissue that they looked at, but we have included other, if there is data out there, we can include it to ourselves. So for example, GTEx doesn't have any fetal tissue in it. It's all, I don't think it even has any kids. I think it's all adults. And of course, one of the things that we were most interested in is fetal brain. But the data exists. People have looked at the expression. People have done RNA-seq and what's thought to be unaffected normal fetal brain. And so we also had a request to add fetal heart to that for our cardiac friends. And so MagicCat actually, also, even though it doesn't come to GTEx, doesn't include those, but for MagicCat, we were able to do the same analysis on the fetal brain and the fetal cardiac data sets and include those as part of our analysis. So if you use MagicCat, it's seamless. You can't tell that they didn't come from GTEx, but they didn't. So if there was a data set like that from CSF, just like we included the fetal brain, we could include that analysis as well.
Great, that sounds fantastic.
Also, we could also help we could, we could have, I'm sure that, we are not the experts in CSF, but if you know that there's a data set and it would be interesting to talk to us about ways to look at splicing and stuff like that, we would be more than happy to do that. That's a whole interesting thing that I never even thought about, so.
All right, were there any other questions at the moment? Paul's put his hand up. Allowed to
So we'll move on.
So
So I'm going to.
I've got one question.
Sorry.
from Paul, I think.
I'm sorry. I misheard. I'm so sorry.
Sorry about that. Thanks, Elizabeth. Just a quick question. These are, I presume, on bulk RNA. Have you done any work on single cell RNA data sets?
That's such a great question. So my understanding is the next stage of GTACs is going to include a lot more single cell RNA. And so we might be able to get much more nuanced, detailed information. And so since we have this, the framework set up, we can basically plug any data sets into it. We actually had one of our immunology collaborators was actually asking about single cell RNA on different sets of immune cells, you know, because we don't know, how well can you use whole blood to substitute for, say, a pure macrophage, you know, culture or something like that, you know. So so it is in conversation. It's not part of MAGIC is all bulk right now, but that is definitely the next thing that's coming. And so we're waiting for GTACs to get there, and then we'll incorporate it.
So if people had single cell data sets, would you be interested in looking at those?
Yeah, so yes, definitely. We can, anyone who is, anyone who wants to share data with us, we can make it part of MAGIC pretty easily. It would be, it would have to be normal, some kind of normal tissue samples.
Yeah. Awesome. I might chat to you later.
Perfect. I would be very, very happy to. Okay, any other? Okay, then let's move on to, now we've we've used all of these technologies, we've made a diagnosis. And so I'm going to talk you through kind of the translational part, how patients come to us to make up the majority of our cohort. So one of the things that we're really interested in, of course, is this novel gene discovery. And we know that exome sequencing and genome sequencing have a relatively low rate of diagnosis. And so one of the things that we've done is create a really enriched cohort of patients that are syndromic craniofacial patients. Although, of course, we're interested in diagnosing children because of their craniofacial anomalies, we also use craniofacial anomalies as a marker of other syndromic features. So, for example, if you have 100 kids with epilepsy, you can do exome and genome sequencing on them, and a large number of those are actually going to have you know, multifactorial. They might have had some hypoxia that was unrecognized, some infectious cause that was unrecognized, and you're never going to be able to find it no matter how much sequencing you do. However, if you take 100 kids that have a craniofacial anomaly and seizures, you're going to get a pretty high hit rate of those that have Mendelian or syndromic disorders. And so what we've done is really focus on these kids that are more likely to have a Mendelian syndrome after they've been through a full evaluation. Pull them in and say, okay, well, you are going to be the highest yield for finding novel disease genes. And a lot of that now is reanalysis of negative clinical sequencing because so many patients get exome sequenced on a clinical basis. Although I should say we're really trying to make this more of a global effort. So we're getting a lot of patients now that don't have access to clinical exome sequencing because there isn't the infrastructure where they are. And so we established this in 2014. We've gotten samples from 23 countries across 6 continents. About half of them are from CHOP, where I work, and about half are external. And we've been really fortunate to partner with a lot of other institutions that are real powerhouses for genomics, and we've learned a lot from them and have shared a lot of our knowledge in doing these reanalyses and these really in-depth exams. And as I mentioned, we're really trying to reach out to patients that don't have a genetics infrastructure where they are. And so we realized that someone can't get an exome because there's no geneticist where they are. There might not be a pediatrician where they are either. So instead of saying, well, you can't be part of the study, we've started actually going to those places. And so I go to different places in the Caribbean, to islands basically where they don't have any geneticists, and see patients there and collect samples, and then collaborate with pediatricians on local pediatricians to get continuity of care and get a diagnosis for these kids. And so an example of that, this is a family that I saw in Grenada, where this child had been brought in. She was below birth weight. She had this really extreme dolichocephaly that you can see. And it turned out when talking to mom that she had an older brother who had exactly the same symptoms, and they had never seen a pediatrician, let alone a geneticist or a neurologist or anything. She was extremely hypotonic. You can see here her, this is not because she's sedated or anything. This is just her resting hypotonic facies. And it turned out that when we sequenced them, in between collecting the samples and giving them a diagnosis, it turns out that the older brother died because they have a mutation in the sodium channel gene that predisposes to cardiac arrhythmias. But now we knew that for the daughter here, and we were able to coordinate with a cardiology group that was flying in to the island and make sure that she now got a cardiology workup, which never would have happened otherwise. And so I think that's a case that illustrates why is a genetic diagnosis important for families, and also just the striking disparities that happen between kids that we see, that I see in Philadelphia versus the ones that we see on our international trips. So this is going back to a family that is from the US and that did have a clinical exome that was done. And this is very typical for patients that we see that have a clinical exome performed, but don't get a diagnosis. And so this is two families that have macrocephaly, which was their craniofacial difference that got them to us. Developmental delay, possible autism. And this is a very classic negative exome. They had a one variant in an autosomal recessive disease. It doesn't fit the phenotype, no candidate genes. Then they ended up finding a random tooth agenesis mutation that had nothing to do with the phenotype too, but nothing here that explained the phenotype. But the pediatrician and the geneticist said, these girls definitely have the same thing, and it's definitely genetic. So they enrolled them in our study. And here's pictures of them. And you can see they're not strikingly craniofacially dysmorphic. And Dong Lee, who's an analyst that works in our lab, reanalyzed this clinical exome and was able to find that they had a splice site variant and a frame shift in this gene TBCK. And long story short, it turns out that this was at the time a novel syndrome, and we were able to collect additional patients. It turns out that unfortunately it's a progressive disorder. You can see here the more severe patients, the older patients require tracheostomy. Basically, they end up with progressive developmental regression, seizures. They are often, we haven't had any patients with the severe form of the disorder that survived past about age 20 or so, and no treatments. Both of the original girls had a milder form because they had a splice site variant. And so this was a milder manifestation of what ended up being a more severe disorder. So we became really interested in this because this really fit our bill for a progressive disorder, a period that we could intervene. Really very little was known about TBCK at the time. It was a newly kind of annotated gene that had very little that was known about it. So I'm going to quickly go through a little bit of this. It turns out that we were able to show that it's a very unusual downregulation of mTOR. The vast majority, I'm sure you know, of mTORopathies involve upregulation of mTOR. So this was kind of the opposite. And so we kind of put together what was known about the domains in TBCK and did some functional testing on patient cells and showed that it's actually a Rab gap protein for Rab7 and Rab5. And that's what leads to the the decreased TBCK. So when you have less TBCK, you can't undergo this appropriate GTP hydrolysis, and you end up with too much active RAB5 and RAB7. And we knew that RAB5 is important for RAB7 and RAB5 are important for mTOR and lysosome formation. And so we were in coordination with one of our collaborators at CHOP, Zilma Ortiz-Gonzalez, showed that that these patients actually have a very dysregulated lysosome autophagosome system, which is likely leading to the progressive symptoms. And it's basically taking one step back, dysregulation of the fed-fasted intercellular system that the cells had. But one of the interesting things that we looked at was that we did RNA-seq and showed that there was, on fibroblast cells, there was actually a common shared pathway with a number of common neurogenetic disorders, Parkinson's, Alzheimer's, Huntington's, and we also picked up this lysosome, which made us think, if we learn about this rare disease, can it tell us about common pathology for more adult onset disorders? We were able to collaborate with Youngshuan Song, also at CHOP, who's an expert in fly neurodegenerative models, and so was able to show that in an RNAi knockdown model that we were seeing dysmorphic neurons. And so this was the first direct proof that this was having an effect directly on neurons and wasn't, say, a metabolic disorder that was just causing this brain differences, something like you'd expect, like OTC disorder or something where there's it actually affects a different tissue, but you see secondary brain dysfunction. We also saw a decreased neuromuscular junction lengths in those flies that was significant. But really, then we got to kind of as far as we could go with the fly model. And so we thought, let's take it to the next step and go to a mouse model. Luckily, we have an excellent core here that is not staffed by cats. And we were able to move forward into a mouse model. And it turns out that the mice are viable, but they do have early mortality, so we weren't able to do extensive testing on them as adults. They resemble the patients in that way. So we did ultrasonic vocalizations that are usually unique syllables that neonatal mice will emit as a result of separation from their mom. And there's a whole, this was so fascinating to learn about, because there's a whole literature about what these different syllables mean and how you can interpret them. And so we started doing this on our TBCK mice. We were able to show that it wasn't just that they were smaller. This is our wild type heterozygous and knockout littermates. And you can see here that these vocalizations are significantly decreased. But it's not just from their small size. They can emit them for as long. The frequency and the duration is the same. So they just choose to make more noises. One of the really interesting things was we went back and it turns out that when we put them on a different background than the adults heterozygotes started, the technicians and the lab members started saying, you know what, these heterozygote mice are acting really weird. I can tell who's a het and who's a wild type in the adults just by how they act, which was very strange to us. We didn't expect that the heterozygotes were going to have a phenotype because the parents had never reported anything. You know, all the parents are going to be heterozygotes, 2/3 of the siblings are going to be heterozygotes, and we had no reason to suspect. But it turns out that we put them through battery testing. They actually had an abnormal acoustic startle response with prepulse inhibition, which could suggest maybe a dopamine dysfunction. And that led us to go back and say, if we go back to our biobank now, do these heterozygotes potentially have a phenotype? So I work within the Center for Applied Genomics at CHOP. We have the largest pediatric biobank in the world that has 800,000 samples. And so it was gorgeously easy for me to go to the team and ask them to do a PheWAS. And we actually ended up getting a hit for looking at TBCK variants and learning disability. So this is where you put in PheWAS, you put in a gene and try to pull out phenotypes from that. And we found in the Hispanic population, which is actually where the main TBCK founder mutation is, that there was a hit for learning disability. And we are certain that that's not being driven by our homozygotes, because no homozygotes are cognitively intact enough that they would be coded as a learning disability. They never learn to talk or walk or be to the place where they'd be diagnosed with a learning disability. So then we thought, well, we also have a biobank across the street at Penn of adult samples that has many, many samples. I think it's about maybe 100,000 samples now, that is linked to the Penn EMR. So we said, hey guys, can you do the same analysis for us? And we were, and the MD PhD I was working with said, oh, I'm so sorry, it's just the stupid toe phenotype, but like, I'm sorry that it didn't work. But we actually thought, but it's, you know, quite a significant hit in the PWAS, and it comes both for acquired hammer toe and also acquired toe deformities. And you can see that we, you know, start reaching significance for hallus valgus and acquired foot deformities as well. And so we're really interested in if this, and remember, this is an adult population. The previous PWAS with learning disabilities was in a pediatric population, and this is exclusively an adult population. So it's not surprising to us that learning disabilities wouldn't come up, because it is extraordinarily rare for adults that have learning disabilities for it to be coded as an ICD-9, 10 in the EMR in the first place. So we are really interested if this is some kind of peripheral neuropathy that's associated with this as the adults age, and that's why we're pulling this out. I'm really interested in you know, if other people have other thoughts on this, but that was completely unexpected and just a really interesting kind of side note that we took. So going further, so really, what is our whole goal here is identify these targets for therapy. So what are the ways that you can do this? Well, you can target downstream signaling pathways. So does pathway-based treatment exist? Well, we'd like it to be accessible, cheap, well-tolerated. A lot of these patients live in countries that don't have well-established medical communities. Wouldn't it be great if it had Amazon free one-day shipping? And so it turns out that you can, as I'm sure you know, you can actually stimulate mTOR in a number of ways. And one of the ways that's been done in many, many, many clinical trials is giving leucine, which costs 7 cents a gram and has free one-day shipping. So we were really interested in this as a potential therapy for these patients. It turns out that after kind of thinking about it and doing more research, we actually were able to find a branched chain amino acid enriched mixture that is mostly leucine, but actually has been maximally formulated to stimulate mTOR. And this is the configuration that we have here. We have shown that it can increase mTOR signaling and control in patient cells. And we started using it in our mouse pilots. Some of the interesting things is we showed that it was, we had a trend toward improvement in our very small pilot studies. We wanted to make sure that we weren't going to cause any off-target effects or anything, so we started in a small thing. We already saw an improvement there. We were also able to show in those mouse brains that it upregulated phospho-X6 as a marker for mTOR, and it strikingly increases autophagic flux only in TBCK mice. So we definitely can't say that's better for the mice, but we're definitely seeing a very discrete expected cellular effect from giving the branched chain amino acid mixture in brain lysates. So here's the autophagic flux data. I'm going to get through some of this so that we have time to talk about the other one. One of the things that we learned is that it turns out that being female is protective in these mice, perhaps in the patients, but definitely in the mice. And we weren't sure if perhaps that was because when we went back into the literature, that it turns out that fasted females have strikingly higher levels of phospho-S6, a marker for mTOR function, at baseline. So we're trying to get all the pieces of the puzzle and put them together, but how, if we know that that's for females, how can we use that to help males and bring the males to the level of the females? And then one of the things that we're really interested in is targeting the expression of this gene. And I think I'm actually going to skip through a little bit of this to make sure that we can get to the next disease that I want to show you about. So I told you we were going to talk about 2 different disorders. Oh, let me pause. And are there any questions about TBCK, any of our treatment ideas, any of the...
So we've got one question in the chat from Paul.
Yes.
Said,
Hit
what
me,
was the
Paul.
basis of choosing to do the vocalization studies in the TBC?
Oh, that's a great question. So we did kind of I didn't present it, but we did kind of a full regular battery of developmental testing, like when did the pinna open up, when did the eyes open. All of those were normal, and we wanted to do something that was kind of the best for looking at neurologic function. And since they start dying, despite our best efforts, around anywhere between, the males die sooner, but they die usually anywhere between 12, really P8 and weaning. We never can wean any of them. So we couldn't do any other behavioral tests on post-weaned mice. And so in talking with the behavioral core, this was what they suggested as the best test as almost a surrogate, I guess, for neurologic function for the mice. But we did do kind of all the regular developmental testing that you do in pre-weaned mice, and all of those were normal, except for the weights and the lengths of the mice, which were significantly decreased. Did that answer the question? Is that, you're saying why do the ultrasonic vocalizations as opposed to adult testing?
I think so. Paul's in the chat, but yeah, I think we're happy with that. Thank you. That's great.
Yeah, and if other people, we still have these mice and we would test them for anything. So if other people have other suggestions for appropriate tests to do on pre-weaned mice, I would be so happy to take suggestions on that, because it is kind of disappointing that we can't do more extensive testing on them. Okay, so let's get to family 2. And this is, actually, can someone remind me of the time? My children have run off with my phone, and so I cannot check to see how we're doing time-wise.
So we're just hitting 52 past, so 8-2.
8-2, okay, perfect. Then I had the feeling that we were getting up on it. So I am just going to tell you very briefly about a new set of mutations that we discovered in histones. And so many genetic disorders are caused by mutations in genes that affect histones. These are these are actually the physical histones themselves. And at this point, we have more than 50 patients that all have missense mutations in histone 3.3, which is encoded for both A and B, all de novo variants. It is also a degenerative disease. They have progressive white matter neuropathy. They get seizures, regression as they get older, movement disorders. This is nowhere near as severe as the TBCK-related. We have patients that are in their 30s, but it's very interesting because there's a wave of regression that seems to happen in the first decade of life. And then there's another wave of regression that happens when the patients hit around 30 years old, where they get maybe like a spastic paraplegia. They've seen a lot of different neurologists, and no one's been able to really define it very well, but it seems like it's common among the 3 patients that we have that are that old. And so these are the faces of those patients. You can see some look quite dysmorphic and some are more typical looking. So I don't think that this is ever going to be picked up clinically, but they are quite severe neurologically. You can see that only 73% that are expected to have achieved independent sitting. Only 37% speak any words, and only 61% achieve independent walking. And the sitting can happen as late as 7 years, and the walking is as late as 8 years. We were really interested in why these missense mutations would cause this. And so we did some advanced molecular modeling with Xavier Cruz at Barcelona, and showed that a lot of these variants affect the different functions of histones. Remember, histones are responsible for wrapping DNA around them, and showed that there's specific cohorts of these mutations that affect the ability of histones to do that. So we are really right in the thick of trying to figure out why these different mutations that affect the histones different ways cause this. There are no loss of function variants. There are no nonsense frame shift, terminations, deletions. There are no mutations like that, either in normal populations or in our patient population. And so when we go through, we were able to show that the cell proliferation is increased in the fibroblasts in these patients, very strikingly. But we're really trying to figure out what the implications of that fibroblast phenotype are for their progressive brain differences. Histones are very important for switching from self-renewal to differentiation. So we're wondering if this is a dysregulation of self-renewal versus differentiation. We do have a zebrafish model that recapitulates. You can see that they do not have as many glia cells, so we're wondering if this is actually more of a glial cell disorder than a straight neurologic disorder. And because it is progressive, we're really excited about looking at known drugs that we have that are able to modify histones, including things like valproic acid, which has already been thought to be used on patients that have germline mutations on these in these genes to help with their glioblastomas. This is one of the major driver genes for pediatric glioblastomas. So we're hoping to, one of the reasons that we picked this to focus on was to piggyback on that work that was already being done in dozens of labs across the country looking at this gene in glioblastomas. So I would love to thank everyone. Of course, this is unbelievably collaborative efforts, and especially Elliot, who has been an amazing long distance mentor. He's a member of my K08 committee on TBCK. He has contributed to the projects, and I cannot say enough kind words about him, both as a collaborator and a mentor. And I would be happy to take any questions in the limited time that we have.
Thank you so much. That was absolutely fantastic, fascinating talk. Do we have any questions at the moment? Feel free to raise your hand or post in the chat. I think Elliot's raised his hand. I don't know how to, I think you're on unmute at the moment, Elliot, if you did want to.
Okay. Elizabeth, fantastic talk. Do you happen to have brain imaging longitudinally on any of those histone patients? And what does it look like?
That's a fantastic question. So we're actually collaborating with Adeline Vanderver, and one of the pediatric neuroradiologists at CHOP to collect that data and to collect the raw images from as many patients as we can. I think we have about 15 patients. Most of it isn't serial. I think there's only 3 patients that we have multiple imaging for. As you can imagine, the patients need to be sedated to get the MRIs. And so it's a little bit of a harder sell for a lot of them. But we are collecting that data, and I don't think a major analysis has been done yet, because we're actually waiting until the original paper was published, and then which is hopefully going to be accepted any minute after being rejected from 14 journals. I think it's finally found a home. And so as soon as that one gets accepted, then that data is going to be, and we're going to close that study, analyze the imaging data, and then publish that as a follow-up paper. So I'm sorry I don't have, it's in process, but I don't have any results for you.
Okay. We can maybe talk offline later.
Totally.
Are there any other questions for Dr. Bhoj? If if not, please join me in thanking Dr. Bhoj for a fantastic talk. That was fascinating, and we are very, very happy you were able to speak to us today. Thank you.
Well, thank you so much. I really appreciate the opportunity. And if anyone, let's see, I think I have I can put my email here. Yeah, if anybody would like to collaborate on any aspect of this, you know, we wouldn't survive without collaboration. And we also have positions open for postdocs if you know anyone who's graduating. I mean, we have positions open for everything, but unlike a lot of people during COVID, we have both the funding and the institutional permission for postdocs. So if you know anyone who wants to come to Philadelphia, we would love to have them.
Thank you so much, Elizabeth. We really appreciate your time.
Thank you.
And expertise. Thanks. Take care, everybody, and watch for next month's announcement.